For so long we talked about NMR methods, various kinds of techniques which have developed over the years, and now we are ready to go into the applications of these methods into the area of structural biology, which is the main theme of this course, NMR in structural biology. So, we have prepared the ground with methods. So, now we are going to see how they can be applied in structural biology. So, therefore, we will have to prepare a little bit of ground for the biology itself. What do we mean by structural biology? What sort of molecules we are going to look at? So, that will be the theme now onwards. So, we are going to spend a little time on introducing the different concepts and different biological molecules. And everybody knows that especially the biologists, they will know that the central dogma in molecular biology is described in this particular manner. Every cell has a DNA which is there inside the nucleus, and this is the hereditary material. So, different individuals have different DNAs. So, therefore, the hereditary comes because of this molecule. So, the offsprings will be similar to the parents, and this is because of the similarity of the DNAs, and therefore, it is in fact used to identify people on the basis of their DNAs. And then DNA is then transcribed into another molecule. The DNA stands for deoxyribonucleic acid. So, nucleic acids are a kind of polymers, and then these ones are transcribed into another kind of a nucleic acid which is called as RNA. This is the ribonucleic acid, and this actually is the process called transcription, and different segments of the DNA are transcribed into a certain molecules RNAs. We will describe that a little bit later, and those segments which are transcribed are called as the genes in the DNA, and then they form RNA, and this comes as what is called as the messenger RNA. This process is known as transcription. So, this process is known as transcription. And from the messenger RNA you have what are called the proteins. So, there is a kind of a code in this which is called as the genetic code, and this genetic code describes what kind of a protein has to be produced. And you know, the proteins are made up of amino acids. There are 20 different types of amino acids, and a set of 3 units of RNA codes for one particular amino acid, and the 20 different amino acids are coded by different codes. These are called as the genetic codes. And then this process of expressing the information in the RNA into the proteins, this is a process which is known as translation. And then proteins are actually the biological machines. Actually, for a long it was thought that proteins are the biological machines, but today we also know that sometimes the RNAs can also be carry out biological functions, but the general belief has been formed, which is true, that proteins are the real biological machines and they carry out the biological functions inside the cell. So, now to explain this a little bit more, so you have the DNA, which is a long polymer, it has more than 1 billion molecular weight. It is a huge molecule DNA. This is a polymer, and it is very long. So, it has approximately 10 to the power 9 units, and these are called as nucleotides. Okay. So, on this DNA there are certain segments, small segments, and these are called as the genes. There is a lot of other things also inside the DNA, and these are the genes, and from the genes you get what is called as the RNA, and this is called as the messenger RNA. There are 3 different type of RNAs. These are messenger RNA, then the tRNA, and then you have the ribosomal RNA. So, if I went to draw the cell here, you have the nucleus, and this contains the DNA, this contains the DNA, and the mRNA and these things that actually happen inside this other thing which is called as the cytoplasm here. So, the genes are expressed in the form of mRNA, that is a particular mRNA. There are other kinds of RNAs which are called tRNA and ribosomal RNA, and tRNA is involved in the translation process from here. Here this is, then it goes to the proteins here. This is the process of translation, and from here to here this is the gene, this is the transcription. And the translation generates the proteins. Okay. So, now ultimately our objective will be to understand the structures and how these processes happen. Of course, this is very complex. There will be many different kinds of proteins involved in every step, and it is an extremely complex process. Clearly there is a kind of a synergy in all these actions of these various proteins and the nucleic acids, and one needs to understand this synergy. In order to understand that, we have to understand the structures and the dynamics of these molecules. So, this actually constitutes the main theme of structural biology, and protein, there are also other molecules inside your cell, and these are small molecules which are the source of energy or metabolites. So, the food you eat is actually degraded into various kinds of small molecules, and these are the markers. You have the ions and things like that. So, but biologically we will be concerned with these macromolecules which are the DNA segments, then you have the RNA segments, then you have the proteins. The ribosomal RNA is present inside what are called as the ribosomes, and these are the machines where actually the proteins are synthesized. So, these are the areas, these are the kind of a powerhouse where the proteins are synthesized. mRNA comes inside the ribosomes, and then there is a tRNA which also plays a role inside there, and the ribosome also contains many proteins. Therefore, the whole thing is a big complex, is a big assembly, and the structure of this ribosome is also an important aspect to study. So, individually one looks at the molecules, the nucleic acid segments, mRNA segments, the tRNA segments, and the proteins. Now, this idea of this process of understanding the structures was of course an important topic, and several people had worked on it, and there are various kinds of empirical rules which had evolved, and on the basis of what is called as a fiber diffraction data, these three people brought out a model of the DNA, the structure. They proposed a model of the DNA structure, and they got the Nobel Prize for that in physiology or medicine in 1962. See, for the discovery of the molecular structure of DNA, which helped solve one of the most important of all biological riddles, that is the replication process. Why is it a hereditary molecule? How is it that the offsprings have similar features as the parents? So, why is the DNA so important? What is the structure? How is the information transferred from the parent to the offsprings, and that is retained in the same manner? Okay, so that was an important riddle in biology, and these people gave an answer to that in the form of the structure of the DNA molecule. So, what did they propose? They showed that the DNA is on the basis of the fiber diffraction data. So they showed that the DNA is actually a double helix. So is wound like this, then you have the another one which is also going parallel. I cannot draw this properly here, but this is a double helical structure which they proposed, and that actually became an important contribution for understanding double helix. There are certain rules as to how the double helix is formed. We are going to look at all of those ones, but they based their arguments on the basis of the fiber diffraction data. Okay, fiber diffraction does not give you such very high resolution. At the atomic resolution it does not give that information. It only gives the kind of a symmetry elements. What are the kind of symmetry elements in the molecular structure? Okay, so either it is proper symmetrical spacings of certain atoms, certain spacing of certain groups, what kind of a structural elements may be present inside the big polymer, because you remember this is a very big polymer, and in this we have these two people, Francis Crick and Maurice Wilkins. These were the crystallographers, the diffraction, the ones we interpreted the fiber diffraction data, and Watson was a chemist who actually built the model of this using ball and stick models. So and then actually turned out to be such an important contribution that they got the Nobel Prize in 1962. Okay. So now we have to go into understanding the structures of these molecules in greater detail. Okay. So before we actually go to that, let us look at these various biological function in a little bit more detail, and that is indicated here. So here, okay, now here also it makes an introduction to NMR, because there is an article written here in Analytical Chemistry for metabolomics and metabolic profiling, and it also has describes what are the various elements in structural biology. So you have the genome to metabolome. So this is the genome, the DNA, that is the DNA which is also called as the genome here, and there are typically about 30,000 genes inside the DNA. In a particular, typically in a living cell there are about 30,000 genes. Now these 30,000 genes they are characteristic of the individuals, okay, that define what is called as the phenotype, and then you have of course phenotype, all the things also contribute to the phenotype. Now this genome is transcribed into what is called as the transcriptome. So you have 30,000 genes which are expressed and produced 30,000 mRNAs. So these are 30,000 mRNAs, this is called as the transcriptome. So this is very characteristic of every individual. Every cell has its own transcriptome, therefore this becomes an important thing to study, what is the process here from here to here. And then from these 30,000 transcriptomes you produce nearly 100,000 proteins. So which means that okay, one mRNA is producing more than one protein. In other words, there is what is called as a mRNA splicing. So therefore different combinations of the mRNAs are made here, and not only they produce individual proteins, but some kind of alterations in the mRNAs and cutting them, joining them together, things like that, and that results in producing nearly 100,000 proteins in a particular cell, and this is called as the proteome. And there are also certain changes happen after the translation, some modifications happen, and those are called the post translational modifications. These are important events that happen in biology, and they are required for particular processes in the biological function. And then from here you have this metabolome. The proteome deals with the metabolome, that is the various kinds of molecules, small molecules which are interacting with this. So the metabolites are those which are the food you eat, which is degraded into small molecules. There are ions, and there are nearly 40,000 small molecules which are actually present in a cell. And these are upregulated, these expression levels are upregulated, downregulated, and things like that, depending upon the disease conditions, depending upon the nature of the individuals. It depends upon the environment, diet, age, lifestyle, drug, disease, and therefore one has to target various molecules in this category. So you can, sometimes you may have to target here, sometimes here, sometimes here, sometimes here, and therefore all this contributes to the research in drug development, disease control, and these are dependent on the environment, the lifestyle, age, etc. So therefore it is a huge topic to study all of these ones. There are many challenges, and of course now the NMR has come of age to provide solutions to many of these situations. So in order to see how one can apply NMR, we will have to go into the details of the individual molecular structures. Okay, so let us begin with the RNA structure or the DNA structure. As I said, the DNA is a polymer. It is made up of nearly 1 billion units, and what are these units? These are called as the nucleotides. So the nucleotide is described in this manner. It is a phosphodiester linkage here. This has the sugar ring here. This is the sugar ring. This is a 5-membered sugar ring, as you can see. This is and then you have a certain kind of a base here. There are 4 different types of bases which we will describe soon, sometimes 5, and if in the DNA has a particular kind of a base called the thymine, whereas the corresponding one in RNA is called uracil. It is, and then you have the phosphate group here, and this phosphate group this continues. This is the monomer unit of the DNA, and this repeats itself. This repeats itself through the polymer. What changes in this? The only thing that changes is this base. So from one unit to another unit, there is a change in the base, nature of the base, and that is called as the DNA sequence. As you go from one unit to another unit, DNA base changes, and that is the one which is the information carrier. The sequence of the bases that are present in the DNA, that is the information carrier, and this has a certain chain direction here. So we represent this as the chain direction going from n minus 1 and n plus 1, it goes in this direction. So therefore we write it as 3 prime to this is the 3 prime end, this is the 5 prime end here. See, these are atoms are labeled in this manner. The sugar ring is labeled in this manner. This atom is called the C1 prime, the C2 prime, C3 prime, C4 prime and C5 prime. And then the C5 prime of this unit is attached to the phosphate group here, this phosphate group, and this phosphate group is attached to the C3 prime end here at this point. Okay, now you see there is an oxygen here. All of them have an oxygen, and this C3 prime attaches through the phosphate group to the 5 prime end of the next nucleotide, and therefore this is the way sequence goes on. Okay, so this is the 5 prime, one end is the 5 prime end, other end is the 3 prime end. The chain goes in this direction, 5 prime end to 3 prime end. Okay, so the nucleotide, one particular nucleotide unit, this is n, this is from one 5 prime end to the other 3 prime end. So include the phosphate group of this into this unit, and the next one of this goes to the next unit. So therefore this is the 5 prime to 3 prime sequence. Typically when you write the DNA sequence, you write in this manner. Okay, so in going from 5 prime to 3 prime, what is the DNA sequence here? You write the various bases which are present in this nucleotide unit, and that is called as the DNA sequence. Okay, so this is the monomer of the entire polymer. There are about 1 billion of these kind of things. Now you can imagine how long this should be. So if you consider the bonds here, number of bonds here, so from here to here how many bonds are there? So this is 1, 2, 3, 4, 5, 6, 7. So these many bonds are there. So typically length, if you consider the length of each bond is approximately, let us say 1.5 angstrom, then the total will be approximately 10 angstroms. From here to here it will be really 10 angstroms. Now the 10 angstroms into 1 billion, so this is 10 to the power 10 angstroms. Then you can imagine how long this will be if you take 10 to the power 10 angstroms. Okay, and how much is 1 angstrom? 1 angstrom is 10 to the power minus 8 centimeters. So this will be 10 to the power 2 centimeters. That is about 1 meter, 10 is about 1 meter, right. So the DNA length will be approximately 10 to the power 2 centimeters if it is completely linear, it goes like that. It is complete, if it is of the 1 meter. However, you see the size of your cell is just about a micron, 10 to the minus 6 centimeters. So then, and then the nucleus is even smaller than that. Now how is this entire DNA packed inside that small space inside the nucleus? So which obviously means that the molecule adopts a very folded structure, all kinds of folding things happen there, and it is completely wrapped around in various ways, and so that it is packed inside the nucleus. And this has to be unpacked when it has to be expressed, when the protein has to be and the mRNA has to be produced, it has to be expressed, so it has to be unpacked and then the proteins have to be produced. Therefore, you can imagine how complex this whole process is, and a lot of research therefore goes on in trying to understand all of this, and how can NMR contribute in this is what we are going to see. Okay. Now this is a little bit of a explanation, is somewhat more detailed at atomic level. So you can see here the atoms are listed here, where 1 prime 2, these are the carbon side, this is the ribose ring, this is the ribose ring here. See the 1 prime, 2 prime, 3 prime, 4 prime, C4 prime, and then the O4 prime, this is an oxygen here. And then at this point you have the 5 prime carbon, carbon is attached to a phosphate group here. So you have a 5 prime OH group, and that corresponds to the phosphate. And the deoxy, this is a deoxyribose. The deoxyribose means that at the C2 prime position here there are 2 hydrogens, okay, these 2 are hydrogens here. Therefore this is the CH2 group. The CH2 group is there is a hydrogen, and the C1 prime, this is actually connected to this oxygen, and this C4 is also connected to this oxygen, and C1 prime here is connected to the base, from here the base goes, okay, and then the C3 prime is connected to an oxygen. And in the case of the RNA there will be a small difference here. In the case of RNA you will see that the 2 prime position here also has an oxygen. So there is 1 hydrogen and 1 oxygen in the case of RNA, that is a ribose nucleic acid, okay. So therefore here in the C2 prime, in the case of DNA there are 2 hydrogens, these are labelled as H2 prime, H2 double prime, and in the case of RNA you only have H2 prime, you do not have H2 double prime, and all of these also have a proton on this. There is also a CH3, on this CH3 there is a proton, on the C3 there is a proton which is called as H3 prime, and on the C1 prime also there is a hydrogen which is called H1 prime, and likewise on the C4 prime there is a hydrogen which is called H4 prime, and on the C5 prime there are 2 hydrogens which are indicated here, and these are the 2 H5 prime and H5 double prime, that is better seen in this picture here. So H5 prime and H5 double prime. So therefore there are a number of protons, so therefore one can study using proton NMR what is the kind of a structure of these individual nucleotides are. Okay. Now what are the bases? As I said, the 1 prime is connected to the base. Now there are 4 different kinds of bases, or there are other 5 here. See, this is adenine, guanine, cytosine and thymine. These are the 4 which are present in DNA. Okay, in RNA thymine is not present, it is replaced by a uracil. Okay. Now you see these are all, these 2 are called purine rings. Okay. So this is a purine structure here. You have a 5-membered ring and a 6-membered ring joined here, this is the purine structure, and these are only the 6-membered rings, these are called as pyrimidines. So the basic unit in organic chemistry, this is the pyrimidine here, and these are 2 purines, and the uracil is also a pyrimidine and it has 2 oxygens here, and same is true with the thymine. The difference between thymine and uracil is that you have a methyl group in the case of a thymine, whereas in uracil there is a hydrogen here. At this point there is a hydrogen. Okay, and all of these there are hydrogens here, in this point there is a hydrogen, here there is a hydrogen, here there is a hydrogen, and so on and so forth. Okay. So these are the structures of the bases, and those are indicated in a color coded manner here. So all the nitrogens are blue here, and the oxygens are in red, and the hydrogens are in white, okay, and the carbons are in green. Notice here there are NH2 groups here, the cytosine has NH2, guanine has NH2 and adenine has NH2, all of these have hydrogens, NH2 groups, and they also have iminoprotons. The guanine has an iminoproton, that is the NH group here, and the thymine also has a NH, uracil also has a NH. Therefore, so far as the nitrogen is concerned, there are amino groups and the imino groups, okay, and there are also some nitrogens which do not have a proton attached, okay. So the cytosine H, and it is at this point that actually these ones are attached to the carbon. This NH is actually will get this hydrogen will disappear and it will be attached to the sugar ring. Similarly, and this will be attached to the sugar ring, base will be attached here and the sugar will be attached here and like that here, okay. So this is how the structure of the DNA and the RNAs are built. So this is the same molecular structure of the bases given in a more explicit manner. Okay. So therefore now this is a kind of a summary of what I explained for so long, and that is the primary structure of the DNA or the RNA. So this sequence goes like this. So as I indicated earlier, so you have a base here and this is the ribose ring or the deoxyribose ring, whatever. In the case of only the carbon skeletons are shown, the protons only are shown for the CH2 groups here, but there are protons here and protons there, those ones are not shown. Okay. So you have the phosphate group here. So therefore this is called as the phosphodiester linkage. We have a phosphodiester linkage in this is called the phosphodiester linkage because there are 2 ester groups, they are phosphodiesters. So every dinucleotide has a particular phosphodiester linkage here. Okay. So you have the CH2, this is the 5 prime end and this is the 1 prime here, 1 prime attached to, you see as I told you earlier, at this nitrogen these bases are attached. Similarly the pyrimidine at this nitrogen the sugar rings are attached, and likewise so this is at this nitrogen the sugar is attached and so on and so forth. So in the purines the sugar is attached to the 5 membered ring, and in the pyrimidines it is attached to the 6 membered ring at this position. And to repeat, you have this 1 prime group here, this is the 2 prime. In the deoxyribose you have 2 hydrogens here and there is 1 hydrogen at the 3 prime, and the 4 prime there is 1 hydrogen here, and this repeats itself in the all along the sequence. So in a linear sequence this is how the structure is made. Now this is shown in a color coded manner here. So where how the DNA sequence is formed, you have the 5 prime phosphate group here, and then there is the base attached to the sugar ring. So the this is called as the backbone. Okay, so the phosphodiester linkage, this is called as the backbone of the DNA. You have the deoxyribose ring here, ribose ring here. So all the ribose ring and the phosphodiester linkages, they constitute what is called as the DNA backbone. And the bases are indicated here C, G, A, T, etc., etc. So this defines the sequence of the DNA, and this is called as the DNA backbone. Okay. Now what these people said, Watson, Crick and Franklin and all of these, they proposed this model that the DNA is not a single strand but it is a double strand, and that is how the hereditary information comes. Hereditary property comes because of this, that is that if you have a DNA and this molecule exists as a double helix, what happens in the double helix? There is one strand runs like this from 5 prime to 3 prime end, and the other strand runs in the opposite direction 5 prime to 3 prime end, and these 2 nucleotide units, the 2 strands are held together by so called hydrogen bonding between the bases, and these are called as the base pairs. So you have the CG pair here, the TA pair here, GC pair here, CG pair here. They also observed that C can pair with G and T with A and no else. Okay. So therefore this is the model which they put up, and of course this was based on previous informations inside available to them, that okay, the number of C's is equal to the number of G's, so the number of T's is equal to the number of A's in the DNA. So on that basis actually they had put up this model. Then you have the C and the G are paired together through hydrogen bonds, and there are 3 hydrogen bonds between C and G, and there are 2 hydrogen bonds between T and A, and so on. So this is how the model goes, that the only certain bases will form pairs. Okay. So now you see, if this DNA breaks apart, suppose you the 2 strands separate out, and in the daughter offsprings if the new DNA is synthesized on the basis of one DNA strand, naturally the other strand which will come will have the complementary sequence. So if the sequence goes C, T, G, C, A, the complementary strand will have to have this sequence here, that T, G, C, A, G, it has to have that. Therefore the information is transferred from one molecule to the other molecule. If this is the parent molecule, during the process of replication, the DNA strands separate out, and each one of them actually become the starting points for the new DNA molecules to be synthesized. When the new molecules are synthesized, they maintain this complementarity of the base sequences, and that is how the offsprings get the same kind of a DNA information, and that is called as the heredity. Okay. Now this is explicitly shown in a little bit more clear manner. The two strands are running in this manner. You see, you have the 5' end of one chain which is going like this, one DNA chain is running like this, and the second DNA chain runs like this. They are intertwined, they are wound together in a kind of an intertwined fashion, and then you have these base pairs that are happening between the two strands. Okay, you have the phosphate backbone and you have the base pairs which are there between two strands which are running in opposite directions. Okay. So this is how the hereditary information comes in the DNA molecule. And to show explicitly what sort of hydrogen bonds are there, and this slide shows you the particular way the hydrogen bonds are formed. A and T, there are 2 hydrogen bonds formed here. This is the thymine iminoprotons and goes here, and the oxygen here goes to the NH2 proton of this of the purine here. The purine has an NH2 here, these are the aminoprotons, and this is the keto group here. This is the oxygen of the thymine which pairs with this, and here is the imino group of thymine pairs with the nitrogen of A, and therefore this is how the hydrogen bonds are formed in this. Similarly, in this case you have the amino of cytosine going to the oxygen of the guanine, and the oxygen of the cytosine goes to the amino of the glycine, and then there is an NHN hydrogen bond. G amino pairs to the nitrogen of the, because you see the color code here. So what is the color code? The blue is indicating the nitrogen, okay, and the red is indicating the oxygen. Therefore these actually clearly tell you what sort of hydrogen bonds are formed between the AT and the GC pairs. So this has been the basic discovery which actually led to the Nobel Prize to Watson, Crick and Wilson. Okay, so we will stop here.